Yes, good evening to the participants. So, welcome to the day 11 session on immunoinformatics and reverse vaccinology. Before stepping on to this today's session, can we recap the last session topics? So, what are the last session topics? Yeah, what are the last session topics? Either you can unmute and answer or you can type the same in the chat box. Yes, we discussed on MD simulation. So in MD simulation, we discussed on especially on a tool called GROMAX. GROMAX is a freeware, open source software, and we have seen the various programs in GROMAX, and we have also discussed on the types of MD simulation. I told you if you have a protein and if you want to check the stability of a protein in different solvents, we can try with MD simulation. And if you want to get an PDB file from X-ray coordinates, we can able to get from MD simulation. And even if you want to check whether your drug is lipophilic and whether I can able to target mitochondria, even this we can able to check with MD simulation. And we have discussed extensively on the ligand receptor interaction, the checking the binding, stability, and the energy of ligand receptor through MD simulation, isn't it? I mean, this I have taken you directly to GROMAX tutorial, and I mentioned you six major steps used for MD simulation. And the analysis we have to analyze through exam grace. This is the tool we have discussed to see the graphs of RMSD, RMSF, okay, radius of gyration. So we have seen even MMPBSA, so energy. So these are some of the results we have discussed in MD simulation. Very good. You recollect this. So, in the I also mentioned the difference between docking and simulation before starting this. That is, docking we generally check the binding affinity of ligand with receptor, but whereas in simulation, so other than checking the binding affinity, we also check the binding stability over a period of time. So we measure from 100 nanoseconds to 500 nanoseconds. Okay, in the publications, good quality publication, they use higher time to check the simulation. Okay, so this is what we have discussed on yesterday. Anything else did we discuss other than MD simulation? So now can I ask you a few questions? Give me two examples of softwares used for MD simulation other than Chromax. I also showed you a few slides related to that. Softwares for MD simulation like Chromax, you have seen. You remember energy minimization methods? So that I have showed you as an example. And also I showed you a few commercial tools. Okay. You have to go through that. Okay. So if you can't able to recollect everything, tomorrow I'll be giving you a break. Tomorrow I'll be giving you a break and we can postpone our tomorrow session to day after tomorrow. Because nowadays even the participant strength is getting reduced drastically, and also the interaction is very less. But very good. The answer is amber, of course. Other than amber, chromos. Can we have a break on tomorrow? Just I'm asking the present participants. Is it okay? So only one participant is answering. How about the others? Can we have break on tomorrow? Yes or no? Because I found so many participants. Very less number of participants. Okay, sir. Because you should have a breathing time, I believe. Okay, if you have a breathing time, like whatever time you have, you can watch the recorded videos. Okay, but instead of completing our entire internship course on 22nd, we can postpone that. Even 22nd, we have uh, mean, according to this course, we have to conduct MCQ. You have to appear for MCQ. We can have 2 or 3 days time for appearing MCQ. Is that okay? That we can postpone. We can complete our 14th session on 22nd. Okay, and the MCQ, which is essential for securing a certificate. The MCQ can be attempted from 22/12/25 to 25/12/25. I can give this much days, 22, 23. So 4 days I've given. So we can complete our 14th session on 22/12/25, and MCQ can be attempted from 22/12 to 25. That is up to Christmas. If you want to complete before Christmas, you can complete it. Okay. So you have a sufficient preparation time also. Just watch the videos and you can do well. But as I earlier mentioned, that the marks are not going to reflect in the certificate. This is just for self-assessment. The MCQ is for self-assessment and also complete the final feedback to secure the certificate. Okay. So our objective is to just gain the knowledge and utilize the skills in research projects. Okay, can be major or minor projects. What you're going to work in future. Okay. So this is the thing. So anyhow, I will be pasting the same message in the WhatsApp, and I inform everyone on tomorrow. Now, let me go ahead with the left out thing. So I asked you about the software tools, what we can use for MD simulation. I got answer like Amber and Chomp, very good. Other than that, we have Chromos, and then what else did we discuss? Yasara, and I think why something. Yasara we have discussed. Desmond, okay. Desmond simulation is popular. Even in Discovery Studio, we have a Discovery Studio simulation. Scroid Engine, we also have a simulation. Okay, I think in Scroid Engine, whatever we use simulation, the name is Desmond. Okay, Gromos Yasara and NAMD, Nano uh, particle related kind of a molecular dynamics. Okay, nanoparticle associated molecular dynamics. So these are some of the tools we have discussed on yesterday. Okay. Expressive cumin for energy minimization. Yes, this for energy minimization. Whatever I mentioned is this for MD simulation. You know, the energy minimization protocol is also same. Whatever mentioned like charm, we have an energy minimization, and we can also do the same even using MD simulation. Okay, now let me take you to the today's session. No, no, we also left something. Whatever we discussed in earlier session. What else? Other than MD simulation, we also discussed on molecular docking analysis, isn't it? The docking analysis, we have worked with Discovery Studio Visualizer. But in the protocol, I showed you Python molecular viewer, but I provided you demo on Discovery Studio Visualizer. Okay. So if you see this uh, today's session contents, we'll be discussing on immunoinformatics, reverse vaccinology. So today we'll be discussing on immunoinformatics, vaccine adjuvants, types of vaccines, reverse vaccinology, case study, indigenous database. That is, we have developed one indigenous database in Sri Venkateswara College of Engineering. We named the database as Immunoanalytical Database. This database was developed by two PG students, okay, under my mentorship as well as my Professor M. Sivanandam sir mentorship. So, and we are also going to discuss on epitope database of B cell and T cell. Now, let me first go with immunoinformatics. What is immunoinformatics? You remember from in the earlier sessions we discussed on chemoinformatics and we discussed on bioinformatics. And a few participants have also given very clear explanation that chemoinformatics, we work informatics on chemical molecules. If we work on biological molecules, that's what bioinformatics is. That answer is perfect. And similarly, what do you mean by immunoinformatics? It means that whatever immunology you work in computational part, that's what we call it as computational immunology. We call this as immunoinformatics, and this is a field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. And if you see this immunoinformatics, basically we study on vaccine and vaccine adjuvants. Okay, vaccine. What is vaccine? A vaccine is a biological preparation that induces acquired immunity against a particular pathogen. In my PhD research, I worked on vaccine adjuvants in detail, and even my present PhD scholars are working on vaccine adjuvants. In future, if you want to work on vaccine adjuvants, especially for the treatment of types of cancer, so you can keep in touch with us. And in our institution, we also have offering PhD scholars with stipend, so you can utilize this, and we can also get plenty of opportunities to apply for sponsorships. Now, let me complete something about this vaccine. I just initiated what is vaccine and defining it. So, vaccine requires adjuvant. So, adjuvant to simulate an effective immune response. The immune response can be a humoral or cellular immunity. Okay, these are the two types of response we can get: humoral response and cellular. The humoral is basically from antibodies, and cellular is from different immune cells. So this is the cellular immunity happens from different immune cells. Okay, so more about this we'll be seeing in the subsequent slides. And if you see adjuvant, what is this adjuvant? Why this vaccine requires an adjuvant? The adjuvant is actually a chemical or a biological agent that is added to a vaccine to boost the same thing, whatever I mentioned, by increasing the efficacy of antigen. If you want to increase the efficacy, obviously we are using adjuvants. Adjuvants can be chemical based. For example, the most popular used adjuvants in most of the vaccine is alum, which is an inorganic salt, inorganic aluminum salt. But nowadays, to reduce the accumulation of inorganic solvents in our body and body fluids, we are using this biological adjuvant. The biological adjuvants can be protein molecules or plant molecules, or it can be the cells itself. I worked on a type of cell called TCs. TCs refers to dendritic cells. The dendritic cell itself can also act like an adjuvant. Okay, adjuvants can be of any types, from chemical, phytochemical compounds, or can be interleukins, cytokines, or it can be the hormones, proteins, and or even the cells can also act like an adjuvant. So the purpose is to increase the efficacy of antigen. Where can we get this antigen? Where can we get these antigens? To know more about this, can we discuss the subsequent slides? Now, I just am explaining you the application that vaccine adjuvants uh, is make antigen more immunogenic. So, if you want to get more immune response, we use that, and this can also use for increasing the antibody. For high antibody production for humoral immunity, we can use adjuvants. And also, it can induce the cellular immunity. So, it means that we generate specific cytotoxic T cells. Cytotoxic T cells are represented by TC, and also can see the memory T cells can also will be increased because of this. So I already come across the vaccine and vaccine adjuvant. Now I give you an example of types of vaccines. Vaccines can be of inactivated vaccines. So examples are hepatitis A, flu, polio, rabies. Live attenuated vaccines, measles, mumps, rubella, smallpox, chickenpox. So can you tell me what is the difference between inactivated vaccines and live attenuated vaccines? Because nowadays Everyone know the difference between a vaccine, medicine, drug. These are the generic terms we use for the treatment of any diseases, isn't it? Can you tell me the difference between these two types of vaccines, inactivated and live attenuated? This is my question. What is the difference between inactivated and live attenuated vaccines? What do you understand? Inactivated. It means that killed vaccines. Live attenuated has live pathogen, but its pathogenicity is removed. But is very good. That's the answer. And similarly, inactivated is mostly killed. Okay. So how we can do inactivation? The inactivation we can use by either treating with chemicals or either we majorly use heat. Just the heat is the cheapest source to kill the viruses. The killed viruses can be used for the treatment of the same viral infection. And sometimes the whatever viruses we make it weak. Okay, so that's what we call it as live attenuated. So this can be also can be used for treatment of the same kind of viruses. Okay, and if you see this messenger RNA vaccines can be used for the treatment of COVID-19. And even if we degrade the viral membranes, okay, we can release the proteins from the viruses. Whatever we release, proteins, these are in the form of subunits. And sometimes we can use the recombinant technology and we can able to increase the vaccine production, especially, even in COVID-19, when we face the pandemic situation. So we want vaccines for more than 8.1 billion people at that time. So, but this three companies from India, they came forward to manufacture as much as possible within a short period, isn't it? So, of course, they have used a recombinant kind of a techniques for the Covishield, Covaxin, etc. Okay, but even here, these subunit vaccines, we can able to use this recombinant technology. So polysaccharide vaccines and conjugate vaccines. So conjugating one protein with polysaccharide, one protein subunit with another subunit. So these are conjugate vaccines. Examples are hepatitis B, human papillomavirus, meningococcal disease. Toxoid vaccines. The toxoids produced from the bacteria can be used for treatment of the pathogens. Okay, so the example is diphtheria, tetanus. If we see any bite from small animals to large animals, obviously the doctors recommend you to take tetanus. Okay, so the tetanus is of course treated through toxoid released from the bacteria. And if you see this viral vector vaccines, COVID-19 is the best example. We have adenovirus vectors and types of vectors, but adenovirus vectors are popularly used for treatment of COVID-19 infection. Peptide based vaccines. The best example is COVAX. So, this is an example of multi-tube peptide based vaccine. Okay. So, we discuss more about this in subsequent slide. Now, let me go to the reverse vaccinology. Classical vaccinology. Everyone know if you want to treat, just now we have also discussed, if you want to treat a viral infection or bacterial infection, or it can be even parasital infection, we can take the same kind of a virus or bacteria. We can use the subunits. We can use the inactivated form or attenuated form. This is what we have discussed. But whatever we discussed, okay, if you want to target to the major population, then obviously we have to scale up. To scale up, obviously we use the fermenters, I mean bioreactors, to increase the production of vaccines. And if you see what is reverse vaccinology, the reverse vaccinology doesn't start with these pathogens like virus, bacteria, parasite. Instead, we start with the existing data available in the form of genome or proteome. So we know that viral genomes and proteomes are already established, viral and bacterial sources. If that are established, you take from that. Don't take from beginning. You just take from already existing. And if you take these kind of a data from any of the omes, genomes, proteomes, and then you start exploring on even structures of the target proteins, and if you and we if we can able to design any kind of biomolecules which can target this structure, then obviously this kind of a concept we called as reverse vaccinology. Instead of taking from the virus or bacteria or parasites, if you go with the databases, that is where you can get information for all these pathogens, whatever I mentioned, specific pathogens, we can also can able to get the structures from the databases, use the structure of targets, and then try to come up with a kind of a vaccine from any of the biological sources. If this is reverse vaccinology, okay, that is what the major immunoinformatics concern. In immunoinformatics, there are types of computational biology, but this is the major thing what the scientists are using. But what are the other kind of computational biology, immunology available? That I'll be telling you. I'll be mentioning on 13th session. Okay. Now let me complete about this. In reverse vaccinology, I told you we start with the genome, then we select the antigen selection. Antigen here I mean the target protein in the pathogen. The target protein in pathogen, okay, that's what I call it as an antigen. So what exactly we want to target in the pathogen, okay? So that will be selecting, and from this we try to identify B cell and T cell epitopes. So after identifying the target, what we do, we try to determine B cell and T cell epitopes. So to distinguish about these epitopes, we'll be discussing in the day 11 and day 12 session. Okay. So whatever we come up with these epitopes, what are epitopes actually? Epitopes are small peptides, which is going to have a length around 8 to 15 amino acids. So whatever proposed length I've provided, okay, based on the types of epitopes, these peptide-based epitopes are used initially to test with small animals. Small animals like rat, mice, and the other rodents like rabbit, etc. So whatever we try to target, so we can able to check the protective immunity, and then we can try with the molecular epidemiology. We can try like based on if you see the viruses or spreading more and expanding with so many population under that conditions. So initially, you should not skip directly to the phase 3 or phase 4 trial, but we have to start with phase 1. That is, phase 1 stands with the mm, healthy volunteers of less population. The less population, it means that we try to test the efficacy of peptide with less than 100 healthy volunteers. If you can able to try with around 1,000 healthy volunteers, okay, that's what we call it as phase 2. So whatever I call phase 1 and phase 2, initially whatever designed peptide epitope, we have to test with healthy volunteers. Later, we can able to check with the diseased volunteers. That's what we call it as phase 3 and phase 4. In phase 3, we try with more than 1,000 people, 1,000 to 10,000 or 1 lakh. It depends on how much population has infected from that particular pathogen. Later, after completing phase 3, we go with phase 4, that is related to FDA approval and post marketing surveillance. So, not only the drugs has to get the FDA approval, vaccines, any type of form of vaccines has also has to be submitted for FDA approval if it is submitted for phase 3 trials. And if you go with the reverse vaccinology approach, the first vaccine ever discussed from the reverse vaccinology is meningococcus B vaccine. This is used for treatment of bacterial meningitis. For treatment of bacterial meningitis, which is actually occurs in brain, Okay, so we can use this meningococcus B vaccine. So how this meningococcus B vaccine is discovered, as I mentioned earlier, the reverse vaccinology, we take a genome, proteome, or even you can take the protein sequences or the structures. So ultimately, we want the 3D structure of proteins, okay, from any of the omics or any of the structure-based depositions. And if you see, we take these structures, okay, target proteins. So initially, they have selected 600 potential vaccine candidates. But when they tried with recombinant technique to scale up, okay, they got only 350. That is, whoever discovered this meningococcus B vaccine. So, out of 600 potential vaccine candidates, that is, we have that much kind of a target proteins in meningococcus B. That is that kind of a bacteria. So, if you see that so initially 600, but it is screened to 350 based on successfully expressed in E. coli, that is successfully undergoing recombinant technique. Later, it is screened to 91. So that is 91 surface proteins only identified out of 350. And from that, only 28 novel proteins have the bactericidal activity. So finally, we have concluded we can able to choose these 28 novel proteins, okay, for the vaccine preparation. So we had all the 28 structures. So whatever peptide we have designed, we can check whether it's going to target in the appropriate binding site or not. Whatever is targeted in the appropriate binding sites, we have freeze that as the most important candidates, and that's what they have used. Got it? So this is how the first ever vaccine came through the reverse vaccinology approach. But nowadays, you can see for many diseases, cancers, even for malaria, and most of the bacteria and viral kind of pathogens. Okay, so we can able to see the vaccine candidates are developed through the reverse vaccinology approach. Now, let me take you on discussion with my publication in the year 2020 on T cell epitope based vaccine design for Nipah virus. So, my team of students have worked on different types of viruses. Okay, they worked on hepatitis B virus, hepatitis C virus. They worked on different novel proteins from SVC as well as from other colleges. The other colleges also are working projects the form of a consultancies, and they completed masters, PhD, etc. under my guidance. Okay, they worked majorly on hepatitis B, C, and Zika virus. Okay, so these are the types of viruses they have worked under my guidance, and for most of them utilize this reverse vaccinology approach. So of course, I told you this approach we can use it for vaccine design. Okay, peptide based vaccine design. And the first project what I initiated during the pandemic situation, actually exactly I didn't started at pandemic. I started at 2019 when 2018 in Kerala, more than 18 people died because of Nipah virus infection. So I got enthusiasm and started working on this Nipah virus. So when I started working on the Nipah virus, I worked on both T cell epitope based vaccine design as well as B cell epitope based vaccine design. Because we have extensive results, I just published the T cell epitope based vaccine. But the one which is most important for Nipah virus is B cell epitope based vaccine, because B cell is the immune cell which can produce antibodies. The antibody therapy is the best for Nipah viruses. But for few viruses, the T cell epitope holds good. Okay, but whatever I found a gap, that I have worked in the yeah, during that time, because most of the target proteins are already worked by the scientist. If already worked, there is no objective, there is no reason to work with the same kind of a protein for publications. Okay, so I identified which only one protein that didn't work. I tried with that, and whatever I published, this research work, computational research work, is taken by a PhD scholar in Pasteur Institute of India. Whatever I worked in computational part, they worked in vitro studies and they come up with a kind of a vaccine candidate. So whatever you acquire in computational skills, and if you try to publish it, it doesn't mean that you have to come up with this. Even one another person, another scholar can follow you, and they can also come up with this. So whatever you do, okay, just do genuinely and present only the genuine results, so that even it won't divert the others take care of in vitro or in vivo studies. I believe she also carried out the in vivo studies. Okay, now let me come across. So also I received the Tamil Nadu state government uh, kind of a grant award in the UG project, and even this project is selected as the finalist for the INA UG project grant award. Only two projects got selected for national award in the India. So ours is one of the project. Okay, and if you see this workflow of Nipah T cell based epitope based vaccine design, I started with a target protein called W. Okay, so the other proteins are already worked by scientists and also published. So just because I had a gap, and of course I found this also one of the important target. I just taken this sequence, and whatever sequence I have taken from Uniprot, but taking a protein sequence, I can use. This is the best website. In Uniprot, we have a database called Uniprot KB, isn't it? In Uniprot KB, you can go and we can take the FASTA format, or even you can use UniPark. The UniProtein archive can give only the FASTA format of the protein sequence. This is the direct thing for FASTA. So whatever I've taken the Nipah virus protein W, okay, I just used a website called a public domain website called IEDB. IEDB stands for Immune Epitope Database. Immune Epitope Database. So this is what we call it as IEDB. Like, however, we find NCBI, which is the major website, which is the largest website for protein sequences, structures, nucleotide sequences, genome, literatures. This is the largest database, no doubt, and this is taken care by USA government through National Institute of Health. And similarly, if you see this, okay, this is a largest bioinformatics database, the NCBI, National Center for Biotechnology Information. And if you see this IEDB, Immune Epitope Database, this is the largest immunoinformatics database. We have plenty of information related to T cell epitopes and B cell epitopes. There are also plenty of tools present in the same website URL to work with these tools and databases. Okay, I have used the IEDB, and other than this IEDB, I have also used other exclusive databases for searching the epitopes. I used more than 4 to 5 databases in this research, and then I compared IEDB with that other databases, whatever I worked. Okay, and whatever I compared, I've screened a one epitope, one best epitope for MHC1 molecule, and another best epitope for MHC2 molecule, each one. Okay, whatever I selected the best epitopes for these two different major histocompatibility complex proteins, which is linked with HLA genes. HLA is the gene and MHC1 is the protein. So I just taken this molecules of human. You know, the MSC molecules are present in antigen presenting cells of human. Antigen presenting cells. So I've taken this antigen presenting cell of human has this MHC molecules, and these MHC molecules can carry the antigen, and this will be presented. Okay, so our objective is whatever peptide we design, whatever peptide we get from the pathogenic protein Nipah virus that acts like an epitope, this we try to bind with the MHC molecules, and then we check whether the binding is significant or not. We report the binding energy score. Whatever we do in molecular talking to check the binding affinity, later I after the molecular talking, so that is the epitope of Nipah virus MHC complex is subjected to, that is epitope MHC molecule complex is subjected to MD simulation. Okay, epitope is from pathogen source, MHC is from the human. Uh, so if you want to use the vaccine to treat human, obviously we have to dock this and that, and this is what subject to simulation. Now, like this, we also worked with SARS-CoV-2. I forgot to mention about this because a team of SVC students worked under my guidance and have secured 3.6 lakhs internship stipend. Okay, and so just because they won a phase one event, they have secured around 3.6 lakhs. Okay, and related to this SARS-CoV-2, because everyone don't forget this SARS-CoV-2. So we can just discuss that in SARS-CoV-2 we have structural proteins like a spike glycoprotein, envelope membrane, and nucleocapsid. Everyone know the spike glycoprotein which is present on the surface is most important target. So even in drug discovery hackathon, one of the hackathon event conducted by CSAR, AACT, they only have sponsored my students for this much amount, and also they have provided an opportunity to undergo internship, online internship in a reputed CSAR institute. Okay, and if you see this kind of research, I majorly worked on this target called spike glycoprotein. Okay, we developed a drugs for the spike glycoprotein. So if you see the structural proteins, we have four numbers. And non-structural proteins, we have 16 numbers. That is non-structural protein 1 to non-structural protein 16. You know, polymerases, proteases, polymerases, proteases, one of the major important targets of most of the viruses. Okay, these are also coming under non-structural proteins. Okay, so we have 16 non-structural proteins. You know why I'm bringing this SARS-CoV-2? I'm bringing just for studying with an example, because whatever peptide-based vaccines we are going to discover from reverse vaccinology. Okay, we have to use it for an application, isn't it? And if you see this COVID-19, that is what they named, because in 2019, this SARS-CoV-2 has spread in most of the population. Okay, and so we call this as a COVID-19 as the disease, and SARS-CoV-2 is the name of the virus. If you say this kind of COVID-19, okay, in India, we are treated with two major vaccines, isn't it? What are the two major vaccines? Live attenuated is a weaker strain. Kindly explain screening process. May I know, ma'am, what screening exactly you're asking? Screening now, what screening? Oh, how to screen. Okay, I understand. The screening process I'll be explaining you on tomorrow. How to screen the T cell epitopes, how to screen the B cell epitopes. This will be uh, discussed in session 11 and session 12. Now I'm giving you some basic idea on that. Okay, now let me take you. Just ask you like what are the major vaccines you have taken for SARS-CoV-2? Very good. Covaxin and Covishield. You know the that SARS-CoV-2 we have more than 100 strains of SARS-CoV-2. So you know that uh, spike glycoprotein. The spike glycoprotein is around 1,300 amino acids, around 1,260 amino acid, I believe. Whatever we have 1,260 amino acid, even if we find a single mutation in that spike glycoprotein amino and protein sequence, obviously it gives a different kind of a strain. So what I want to mean is we have more than 100 mutations, around 100 mutations in spike glycoprotein of SARS-CoV-2. Each and every mutation is leading to for it different strains. But if you see Nipah virus, we don't have any mutation. We have only one Nipah virus. But in SARS-CoV-2, we have 100 SARS-CoV-2 viruses. So the treatment is very difficult. But initially, whatever we faced in 2019 is a major, is a basic type of SARS-CoV-2 virus. Only for that one strain, for the initial strain, whatever we got infected, for that, the Covaxin and Covishield is used for treatment of that infection. You know that we can't able to use this Covaxin and Covishield for the other 99 SARS-CoV-2 viruses. Okay. So then, if Covaxin and Covishield is used for the initial SARS-CoV-2 strain, I'm asking you the next question is, what is the efficacy of these vaccines? These vaccines has to be given in two doses. So first we have to give first booster dose and second booster dose. Okay, you remember that? For Covishield, we should take minimum 60 days and Covaxin minimum 30 days to 45 days. Some kind of time period is there, isn't it? So after the first booster, we have to take even the second booster, or else this won't work. That's what you have heard. What is the reason? To increase the efficacy, we are using this. What about third booster dose? This is not actually essential. But the what is that? Uh, if we take a vaccine, we have some shell life, isn't it? That is for if you take the SARS-CoV-2 vaccine, okay, this has to be taken for each and every year. The SARS-CoV-2 vaccine has to be taken for each and every year. If you take the polio vaccine, we can take only once in the lifetime, isn't it? But we can't take SARS-CoV-2 vaccine like that. For each and every year, we have to take. So there is no necessity to come up with third booster. If you start with third booster, it will be like a first booster for the second year. Got it? So that's a fact actually for SARS-CoV-2 infections. Okay, now let me discuss. I just asked you, what is the efficacy of these vaccines? That is after the second booster dose for the initial strain of SARS-CoV-2. This can't be used for the other 99 types. Maybe it works for few types, maybe not more than 10. The COVID shield after the second dose can give uh, not more than 83 percentage efficacy. Covishield showed not more than 83% efficacy, and Covaxin is around 91 to 92% efficacy. I can show you the maximum. That is only 92% patients are getting treated out of 100 patients. This is what I call it as efficacy. Covishield are going to treat only 83. 17 are not getting cured. So what is going to happen? Fatality. Okay, so you can see bulk fatality happened, and many patients, those who got lung infection with SARS-CoV-2, they died, isn't it? So this is the efficacy even for the initial strain of SARS-CoV-2. That too you have to note. But if you see the subsequent strains, you have alpha, beta, gamma, delta, iota. You have plenty of strains of SARS-CoV-2. For these strains, for few strains, this Covishield and Covaxin is less than 15 percentage, less than in some less than 13 percentage. 13 percentage. It means that the remaining 87 has to die. Very bad, isn't it? So for each and every SARS-CoV-2 strain, we can also use the reverse vaccinology. Okay, this can also be used to come up with. So we can come up with a new kind of a vaccine. You know, so why I brought all this? So to discuss just about a vaccine called COVAX. There is a vaccine called COVAX. Did you hear about this vaccine? We heard only Covishield and Covaxin. Also, some might have heard about Sputnik. We call this as a Russian-based vaccine, isn't it? So you might heard about these three vaccines alone in India. But you know how many vaccines came in 2020, 2020 for the treatment of SARS-CoV-2 infection? How many vaccines are available for treatment of SARS-CoV-2 infection in 2020? How many vaccines? 89 vaccines. Okay, around 89 vaccines came in 2020. We had very less time, you know, because the world is declared pandemic in April 2020. Sorry, April 2019. And from 2019, so after the computational vaccine discovery, design and discovery, we have to test these vaccines in vitro studies. Then we have to come up with in vivo studies with small animals. Then only we can try with phase one, phase two, phase three. So if we are infected with SARS-CoV-2 infection, we are in phase three patient. I mean, isn't it disease patient? So it took not more than 1.5 years to come up with these kind of vaccines in the market. But even fewer successful, fewer unsuccessful. And if you see this Covax, you know how much successful it is at that time. It is more than 99.9 percentage successful. This showed this much efficacy. This Covax is a multitope based vaccine. It means that multiple epitopes are tagged together, and you know whatever I call it as a Covax, this is available only in US.
Okay.
This is a USA based vaccine, and this USA based vaccine, and this showed more than 99% efficacy. So only I'm bringing this importance. Even though we have RNA vaccines, recombinant vaccines, why we have to bring this kind of peptide-based vaccine? The best example you can find here. If you see this, we have a T helper epitope, cytotoxic T lymphocyte epitopes of MSC1 and MSC2, and CPG, and this also tagged with one receptor binding domain of structural fragment of spike glycoprotein. So we have multiple peptides linked. Okay. And this is a universal peptide. Like how you find a universal primer, we have a universal peptide. And these peptides are specific to the viruses. And then also it is tagged with adjuvant. The adjuvant is a helper T2 cell adjuvant. That is a kind of aluminum phosphate. Okay. So this kind of an constructor, what we call it as a COVAX, showed more efficacy. If you see these vaccines, okay, the safety profile, that is whatever peptide based vaccine, this showed excellent profile with respect to safety. And immune tolerance is also very good. And you can see even convenience is also very easier to use. Commercialization is stable, simple, cost effective. When compared to monoclonal antibodies, which is more expensive, and this is not even more specific when compared to monoclonal antibodies, this COVAX is more specific in treatment of SARS-CoV-2 infection. So, like this, we can see plenty of advantages in COVAX vaccines. So, we can see this types of vaccines, T cell epitope based vaccine. So, we can able to see the T cell based epitope based vaccine have two types. One is MSC1 molecule and the other is the MSC2 molecule based vaccine. Okay, so we have two types of T cells like helper T cell and cytotoxic T cell. Based on this, we can able to get two different types of epitopes. So more about this, of course, we'll be discussing it on tomorrow. B cell epitope based vaccine. So in B cell epitope based vaccine, in antibody we can able to see an antigenic epitope, and this is what we can able to see. This epitope can be classified into two types. The one what is the linear epitope, which is a continuous residues linear epitope. The other is a discontinuous residues conformational epitope. So these are the two types of B cell epitopes. Receptors, you can see the types of receptors, T cell and B cell receptors, distinguishes with respect to structure. In T cell, we can find this receptor, T cell receptor, and B cell receptor. So related to the helper T cell. Okay, this helper T cell, okay, is going to interact. Is going to have a cluster differentiation molecule called CD4. The CD4 of a T cell is going to interact with MHC2 of antigen presenting cell. So, to know more about this kind of an immunoinformatics, a kind of a vaccine design, one should have a knowledge on immunology. So, can you tell me what are the examples of APC? So, often I am showing you APC, APC. What are the examples of antigen presenting cells? What are the examples of antigen presenting cells? Yes, very good. Dendritic cells, macrophages, B cells. Superb. He also brought the B cells. So, all these cells, B cell, dendritic cell, macrophage, interact with T cell. And if this cell interact with T cell, the T cell gets activated. And one T cell is going to proliferate into two T cells. Okay, so just because this T cell is going to interact with the antigen presenting cell, I told you if you have a dendritic cell, this dendritic cell is going to have a receptor, and this receptor, if it is binding with the T cell receptor, then obviously the T cell is going to proliferate into two T cells. And these T cells further take care of the cellular immunity. And if this T cells interact with B cells, it can also produce tumoral immunity by the production of antibodies. So CD4 T cell interact, that's what we call it as helper T cell, interact with MSC2 of APC, and cytotoxic T cell denoted by TC interact with the MHC1 of APC. There is a slight difference in MHC1 and MHC2 structure of APC. So some APCs has only MHC2 and some has MHC1, and few APC has both the molecules. And if this molecule, if this cell and the corresponding receptor, surface receptor interact with these molecules, this is going to happen. Activation followed by proliferation. So dendritic cell markers. So in my PhD research, I worked on extensively on dendritic cell markers. I found out in human dendritic cells we have around 162 markers. Out of 162 markers, the most important markers to measure the T cell activation is CD80, okay, CD80 and CD86 because these are co-stimulatory molecules. These co-stimulatory molecules interact with CD28 of T cell. In 2014, Ralph Steinman, okay, from Rockefeller Institute, has received Nobel Prize in 2014 who worked on dendritic cells. And similarly, I believe in 2017, those who worked on T cell activation, and especially those who worked on activation as well as T cell suppression, okay, they have received on 2017. Like this, you can see many scientists, those who worked related to this kind of immunology, received subsequent Nobel Prizes. Of course, this concept is essential to know about the treatment of cancer as well as the autoimmune diseases. And other than these co-stimulatory molecules, of course, I mentioned you earlier, the interaction of MSC1 or 2 with CD8 or CD4 can able to influence the activation. So just the CD8086 binding is not sufficient. Along with the CD8086, even this MSC molecule has to bind. And even if the T cell receptor is also going to interact with the MSC, okay, then obviously T cell receptor is also going to interact with the MSC. Then obviously this three molecule interaction is sufficient for stimulation of dendritic cells. Okay, we have plenty of receptors, but these three molecules are most important for activation. And if you see this MHC, I told you we have types of MHC, MHC1 and MHC2. So this is an example of MHC1. You know, MHC1 kind of an activation happens through MHC1 pathway. So here, one of the virus or bacteria is getting inside, and if it is degraded through proteasome, these proteins are split into various peptides. These are transported through transported associated protein, and whatever peptides pass through the transported associated proteins cross through these proteins, okay, ERAP, and then you can see the MSC molecules are present initially in this endoplasmic reticulum. So whatever we find this MHC1 present in endoplasmic reticulum or transported through Golgi apparatus, Golgi bodies, and it is further transported through the secretory vesicles, and the MSC molecules are present in the surface or exposed to the surface. Whatever MSC molecule we have, and the antigen peptide which is present at the top. Okay, that's what we got from the viral pathogen or bacterial pathogen, which is getting inside our immune cell. Okay, so that thing, that part of a peptide, I mean, a part of a viral protein is going to present with the T cell receptors. So if it's going to present with the T cell receptor, obviously you can find an immunity is generated. The type of immunity what we generate is a kind of a cellular immunity. So these cells, whatever acquired the immunity, can combat directly the viruses. You got it? So to combat a viral infection, so this is the mechanism of MHC1 pathway. So structure of MHC molecules is shown here. MHC genes and functions. Okay, we have types of MHCs as I earlier mentioned. Class 1 MHC, Class 2 MHC. Class 1 MHC is something related to cytotoxic T cells, and class 2 MHC genes encode glycoproteins related to helper T cells. Class 3 MHC genes are something related to complementary proteins. There are many differences between the MHC 1 and 2. To know more about this, study from any of the immunological textbooks by Reut and Mail to be etc. Okay. So MSC molecules, one we should remember that in MSC molecules, actually we have genes. The MSC class 1 has HLA-A, HLA-B and HLA-C. If these are the MSC1 genes, MSC2 genes are majorly MSC, DP, DQ and DR, and the location of these genes in the locus of the chromosome is mentioned here. Okay, so these are the major MHC class 1 genes as I mentioned, and these are the major class 2 genes. But we also have minor MHC class 1 and class 2 genes. These are the minor genes. Minor genes are considered the minor importance. And this is the major database we use, immunological immune epitope database, which is the most important immunological database. So I think we have discussed that earlier that biomolecules and even plant molecules and even dendritic cells can also be used as an adjuvant. So in this AH03, AH01, so plant-based saponins can be used as an adjuvant. The saponins are also used as an adjuvant for preparation of or manufacture of SARS-CoV-2 vaccines. In my lab, okay, we worked on IL-2 adjuvant, IL-4 adjuvant, GM-CSF as an adjuvant, that is granulocyte macrophage colony stimulating factor, interleukin 4, And we also worked dendritic cell as an adjuvant. Okay, and also we tried the plant-based compounds as an adjuvant. So whatever the recent trends, contemporary researchers on the adjuvants, we also worked in our institution. So especially in my research, I worked on Vaccinology, a steroidal lactone, and also worked with Perivinon B, a retinoid from a plant called Perivia diffusa. In my lab, we have developed even a database. Okay, that is with C Sharp, SQL, and PHP. So for plant-based adjuvants, we got around 1,000 interactions from Discovery Studio. We had a commercial package called Discovery Studio. We worked with 1,000 kind of a talking, and whatever folders and files we had, we just imported in this online kind of a web interface, and then whatever we uploaded here, we can able to see the best adjuvant for any diseases. For example, if you want to search a adjuvant for an asthma, we can able to find which is the best adjuvant. And if you want to check for even cancer, we can able to get a solution for the cancer. So we have worked extensively on the plant-based adjuvants for the treatment of almost all the types of cancer and autoimmune diseases. So this is a part of my PhD research. Whatever plant molecules we take, okay, these plant molecules can be used to differentiate monocyte to immature dendritic cell, and this can also be used for maturation of dendritic cells. So I just need a minute to complete. So monocyte to immature, immature to mature dendritic cells, the plant-based compounds can be used as an drug adjuvant. So this is a kind of a thing I used for treatment. Okay, so I've completed this session today. So more about the practical part, how to work with the T cell based epitope based vaccine. We'll be discussing on tomorrow, but this you can able to go through meanwhile. So I'll be going extensively on the usage of IEDB website. iedb.org is the URL. Here in this, you can able to see disease information. Like if you want to work with allergic disease, autoimmune disease, you can find the information here. Okay, like if you type the asthma, you can see what are the types of peptides we can use for treatment of asthma. Like this, we can go with any a number of diseases. And similarly, if you want to come up with a kind of a peptide-based vaccine candidate for human, mice, and even dog, camel, non-human primates, we can able to find it from here. Even we can find the peptide-based vaccines for the wild animals. Okay, so and for T cell based epitope prediction, we can use these tools present. And if for B cell, we can find this information. These are the tools used for B cell epitope. And overall, epitope analysis tools can be found from this. And this is the summary statistics. So one can able to explore in this, okay, if you have a time. So more about the usage of IEDB and other tools like ProPred. ProPred is an indigenous server from our country. That is from IIT Delhi. A professor called Raghava has developed so many tools for immunoinformatics. One such tool is ProPred, which is an MSC class 2 binding peptide server. Okay, so this is a T cell epitope prediction server. Like this, I have developed even for B cell and other kind of an applications. So to discuss more about these kind of servers, okay, an Indian-based server and the most popular server of immunoinformatics database, we'll be discussing on Saturday. That is on 20th. Okay, so let's have a break just because the strength is getting reduced gradually and even interaction is reducing. So I give you a day break to acquire this. Okay, so thank you very much for your patience, interaction, and giving the suggestions, valuable suggestions. Okay, any doubts you can ask me, the others can leave. If no doubts, and if you're accumulating any doubts, you can also ask me in the next session. Have a great day to you all.